This is Steve Saltwick, and welcome back to Your Brain, a Personal Tour. Today's seminar is about the decision to act. Whereas Hamlet had it easy, he only had to make a decision about whether to revenge his father or not. In everyday life, the decision to act is much more complicated. We're going to talk about the 4 ways your brain makes a decision. Some are pre-wired by Mother Nature. Some rely on mental time travel. And some are so habitual, we don't even think about them. Each type gives us unique insights into our brain. So I hope you enjoy the seminar. Let's begin.
La noche irá sin prisa de nostalgia. Habrá de ser un tango nuestra herida, un acordeón sangriento nuestras almas. Seremos esta noche todo el día.
I want to frame the next 2 seminars with an analogy. Imagine you're walking in the woods and you come to a fork in the path. Of course, you want to take the path less traveled. So, how does your brain decide which path to take? That's the topic for today. Then the topic next week is, once your brain has made a decision, how do you remember which path you took? That is the topic of Seminar 3. I've added 2 new books to the reading list. The first, Making Up the Mind, is a good general text for further understanding of how our brains construct our mental world, including how different parts of the brain, say the visual and the movement parts, may have different opinions on what the real world is all about. The second book, The Brain from the Inside Out, is a more technical book. I suggest you need some background in biology and other science disciplines to fully appreciate the text. This book, however, is written by a pioneer in the field and represents some brilliant and thought-provoking frameworks for understanding how the brain works in making decisions. Let's get back to the background of the opening quote. Amelia was the first female soloist across the Atlantic. She was a best-selling book author, married to a famous book publisher, George Putnam. She was also a strong advocate for equal rights. Fierce determination marked Amelia once she had made a decision. That's why she's on the slide today. She tragically died in 1937 on a circumnavigation flight around the planet due to a memory problem. But memories are the next seminar. We'll talk about decisions today. While Hamlet had it easy, life presents us with everyday demands that require decisions, and they begin at a very early age. Let's take a look at some examples.
The marshmallow test is a really great way to show how children delay gratification. We tried it out with the 4 children we've been following since September 2010, Alfie, Millie, Mackay, and Pratmesh. Here's how it works. We had each child on their own sit at the table at a desk with a plate and 1 marshmallow. They could either choose to eat the marshmallow, the 1 marshmallow right then and there, or they could wait until I came back into the room and have 2 marshmallows. I left them alone in the room for 15 minutes. Take a look. The marshmallow test has been used for decades by psychologists. It's been used with children to predict later academic success, including literacy, SAT scores, and other academic outcomes. There's no definitive answers from the marshmallow test. It's not a matter of passing or failing. What we're looking for is whether children can really resist this piece of white candy sitting in front of them that's sweet, that, the smell of it, the allure of the marshmallow. In Pratmesh's case, we really saw this added curiosity because he had never actually tasted a marshmallow before. All of the children managed to show some level of self-control and resist the temptation to eat the whole marshmallow. As you can see from the footage, you can catch a glimpse into children's ability to control their impulses. This ability, which is developed around the time of kindergarten, can be linked to other outcomes later in life. At the end, the marshmallows were in kind of different states. Some had been squished, ripped apart, nibbled around. There was this temptation and there was this impulse to kind of try it out.
This video is typically presented as an example of the delay of gratification, or an exercise in self-control. I think now that you've seen what the children actually did, you'll recognize that they decided not to eat the marshmallow by deciding to do something else. Much of the socialization choices we are presented with in life are not simple delays of gratification. It's deciding to do one set of actions rather than another. I said there were 4 ways of such decisions being made. Let's look at each one in turn. The first kind of decision is a reflex. It's a decision that Mother Nature has pre-wired into your brain, because this decision is too important for your brain to deliberate. It's a stereotypical behavior, typically of defensive nature, and it's designed to protect you, to keep you alive. But don't let the idea that only simple movements are reflexes. They can be quite complex. They can be actually whole body sometimes. In this clip, we'll see how an enterprising veterinarian uses a binder clip to freeze and calm a cat. The effect is dramatic and harmless. The cat is not traumatized in any way. The reason this hack works is that it activates a reflex in the cat from his kitten days, when he was carried by his mom by the scruff of his neck. Now, my wife and I are dog people. Do not try this on a dog. But for cat people in the audience, I'd love to hear your experiences with trying this. Reflexes can even be moral judgments. Here we study the actions of 6-month-old infants to the actions of geometric shapes. The shapes must have eyes for this experiment. That's important. The infants watched a plucky circle trying to scale a mountain. A triangle, not shown, helped the circle get to the top, but a mean old blue square pushed the circle back down the hill. Infants reliably reached for the helper geometric shapes, the triangle, rather than the hinderer geometric shapes, the square, thus performing what many would say is a moral judgment of the character of the shapes. But wait, it's a little more complicated than that. In this experiment, 9 to 14-month-old infants were observed to record their preferences to either graham crackers or green beans. I know what you're thinking, but 47% of the 9-month-olds preferred green beans. Then the infants observed 2 hand puppets express their food preferences, crackers versus beans. When the infants were then given a choice of reaching for one of the puppets, they invariably chose the puppet with the same food preference. In other words, they preferred the us of a puppet liking a similar food. Babies also love to see a food us being helped, and conversely, a food them being hindered. So this is not about infants preferring helpers. It's about them preferring agents that are similar to them and preferring agents that hinder puppets that are different from them. So how to make sense of these somewhat surprising results on simple reflexes? The theory I subscribe to is that Mother Nature endows us with pre-wired emotional reflexes, kind of like the automatic mode on a camera. A simple, quick action that's appropriate in maybe 80% of the situations. As we age, we encounter more complex social situations. Our brains have evolved more advanced systems, like the manual mode of a camera. The advanced systems are very good, but they must be consciously and deliberately applied. We'll study those in just a minute. So let's look at Homer and take a crack at summarizing this section. The first thing Homer says is that reflexes are a do-it-now. They are time-urgent, no time for deliberation. They're very useful in avoiding or reacting to harm or threats with an automatic range of behaviors. And those behaviors can be as simple as a knee jerk and as complex as a moral judgment. Reflexes most probably underlie us versus them decisions that may have been useful in our evolution but may be out of place in the modern world. Let's take a look at our second type of decision. We'll call such a decision an associative decision. These decisions unleash a reflex-like behavior, but from a learned association, where that association is a mental connection formed from learning. The science of this learned association is uniquely associated with Ivan Petrovich Pavlov, shown with the white beard here. Dogs were measured in their saliva response to food, which reliably elicited salivation, and to a lighter tone, which did not elicit salivation. When the lighter tone predicted the delivery of food, dogs salivated to the light, a learned association that drove a decision. Such learning is often called classical conditioning or Pavlovian conditioning, and the decisions are termed associative decisions. They represent a fundamental type of decision making your brain and are actively studying today. I must also note that my wife Becky is a professional dog trainer, so I know she'll be very proud of me for getting a dog into these slides. The dog of research today is the California sea hare. You see two of them here. These animals have very simple behaviors. They basically move around in the sand, they squish water with a siphon, you'll see it disturb the surface of the water, and they use a gill to breathe. These animals are the perfect research animals for this type of decision learning. They have illuminated the exact mechanism of how all brains learn such a behavior. And they've even earned a Nobel Prize in the process. Well, not these two hares. The scientist Eric Kandel, who conducted the research, got the prize. The scientific name for this dude is Aplysia californica, and he's cool for one unique aspect of his brain. His brain only has about 20,000 cells, compared to the 86 billion in your brain, and they are huge. Many are visible to the naked eye. Neuron R2 is over 1 millimeter in diameter. It's roughly the size of the B on the Liberty penny. If you're too young to know what a penny is, Google it. These individual neurons can be uniquely identified visually. That means we can experiment with multiple different California sea hares, but study the same neuron, as they learn and express an associative decision. Let's teach this dude a new trick and see what happens in his brain. And we're going to focus just on a few neurons in his brain. We're going to use two stimuli. One will reflexively cause a behavior and is called an unconditioned stimulus, or US for short. In this experiment, it's a mild shock to the animal. Our guy will reliably retract his siphon to this US stimulus. The other stimulus is touching the animal. Normally, nothing would happen. The guy would leave his siphon out and about. But if we pair that touch, in this instance called the conditioned stimulus, or CS, with a US of shock, we find that after training the animal for a while, there's a vigorous withdrawal of the symptom when we touch him. The animal has decided to withdraw his siphon since it's associated with the shock. Since we can see his brain cells, and we're going to focus on this connection of those cells, we can watch and see what happens exactly in terms of the changes underpinning this change in behavior. Now, when we expand our view of the connections between the sensory neuron and the motor neuron, what we see is the creation of a new path in the brain. This path is a combination of two changes. There's a short-term change where previously empty legs of the sensory neuron become filled with additional packets of neurotransmitter, C1 in this diagram. Now, all of a sudden, the motor neuron is affected by the sensory neuron and the siphon begins to withdraw. Over the course of about 12 to 18 hours, a dramatic event happens. There's actually growth in the brain denoted by number 2. New legs appear in the sensory neuron and they become populated with neurotransmitter. There's a long-term structural change in the brain that reflects this learning. We say it's a memory. So the short term is a chemical change by populating empty synaptic terminals, and long term, in this instance after 24 hours, right, there's permanent structural change in the brain. Wow. Now note that that new growth takes up space in the brain. And that's okay if you're a squishy sea hare, but what about a human brain? Over the course of the first 3 years of life, your skull almost triples in size. I mean, that's space for a lot of learning, as any parent or grandparent could tell you. But after that, the skull size is relatively constant. So then, how do we learn new things if there's no room in the skull for increased brain circuit? I mean, something's got to give, right? Well, that's a problem for memory, and we're going to look at that in detail in the next seminar. These same kind of changes happen in other organisms, like rats, and like us. These kinds of changes are important to understand. Let's take an example. We already know that a rat will freeze if you threaten her, say with coyote urine. She'll also freeze if you shock her foot. The pathway in the brain for that foot shock is shown right under the US label in this slide. Sensations for the shock go through a couple of areas of your brain and then concentrates into an area of the brain called the amygdala. The amygdala is a specialized structure in mammalian brains that detect, among other things, items that represent a threat to the organization. When a rat gets a foot shock, the sensation travels through her brain to the amygdala, which drives the behavior of freezing. But freezing isn't the only thing the rat does. There's also internal changes. Her blood pressure goes up. Stress hormones are released into her system. And after we associate a tone with such a shock, the tone will elicit the external behavior of freezing, but also the changes in the internal state, rising blood pressure, release of stress hormones, etc. In essence, the rat has an emotional response to the tone, fear. This concept of emotion can be used for the treatment of others. It turns out about 20% of the troops returning from Afghanistan or Iraq suffered from PTSD. Standard therapies only resulted in about 60% remission. Many members of the Special Forces especially reported hypervigilance and anxiety, similar to the effects shown in the movie American Sniper. Well, if you block a specific autonomic nerve center, the stellate ganglion, with a pain blocker, there are widespread reports of immediate and profound PTSD relief. One shot seems to block the emotion-generating aspects of high heart rate, fast breathing, and muscle tension. In other words, blocking the physical attributes that underlie the emotion seems to release the perception of stress and allows therapy to progress. More recent research seems to indicate that the stellate ganglion blocks can even be effective at the relief of certain impacts from long-term COVID. In other words, for example, the loss of smell. So time to ask Homer what he thinks about this whole set of associative decisions. And Homer has one major point to make. And that is associative decisions change your brain. And it changes your brain with new growth. That structural change as you learn things has several consequences. As we know, your brain is fully encased in bone, which means it's within a fixed volume. If every time you learn something, there's new growth in your brain, sooner or later it's going to run out of room. So how does the brain deal with that? It turns out this is one of the prime functions of sleep. But right now, we're going to explore the next kind of decision-making, the most complex decision-making your brain can do. These kinds of decisions are called deliberative decisions. They are decisions where we have to trade off priorities or options. We could do one thing or the other. We could evaluate the options before we actually decide and then commit to a particular behavior of action. Believe it or not, even our little friend the rat here goes through the same events in his brain. We're going to see a rat's brain go through what can be thought of as mental time travel as he makes a deliberative decision. And what goes on in a rat brain goes on in your brain. Both our brain and the rat brain have structures that help us perform these kinds of activities to really get a good handle. And to really get a good handle on where we're going with this, we have to look at a couple of different places. So let's take them one by one. The first brain area I want to talk about is called the hippocampus. We see it here in a rotating model of your brain. It's the highlighted linear structure at the base of each hemisphere. It's about the size of your little finger, and it's amazing that such a small part of your brain is so important in the way you make decisions. The hippocampus came to prominence when this young man, age 27, received an operation in 1953 to cure his epilepsy. The operation removed his hippocampi and other parts of his temporal lobe. The operation was very successful in curing his epilepsy, but it came at great cost. In essence, this person lost the ability to form new memories as a result. While he was alive, he was known as H.M. He died in the early 2000s, and we now know his real name, Henry Molaison. Henry was well aware of his memories of childhood, but every time he met his doctor, he had to be reintroduced. He'd remember the doctor's name during the course of the session, but the next day he had to be reintroduced all over again. Henry could make certain memories. For example, he could learn drawing and coloring tasks, so he had procedural memory or motor memory. But making memories about his experience, something called episodic memory, and using those memories to make decisions was way beyond Henry's capabilities. Events like these brought the hippocampus into the crosshairs of intensive research. The hippocampus had been known to neuroanatomists since the 16th century. In fact, the first guy that described the hippocampus actually thought it looked like a silkworm. The hippocampus from a human brain is shown on the left, and a nice little cuddly seahorse is shown on the right. The neuroanatomist changed his mind and said after further inspection, it does look like a seahorse. So being a good neuroanatomist, he formed a name from the Greek, and voila, we get a new name for a very, very important part of your brain. Fairly recently, there's been a major discovery about the hippocampus, and that is the discovery of hippocampal place cells. These are cells which are like GPS cells. After a rat has explored some space like this S-shaped track here, you'll find brain cells that are active when the rat is at a particular point in space. For example, if we record neurons in this rat's brain and we let him run down this track, when he gets to the yellow section of the track, we would see specific neurons become active. The neuron's burst of activity might sound like this. So there would be a population of cells within the rat's hippocampus that would preferentially burst when the rat was at a particular place. When he was at a different place, say the orange part of the grill or the maze, a different hippocampal place cell would fire. By convention, we usually color the neuron's activity. They're not really yellow or orange, but it helps our understanding of what we do, of what they do, if we color them uniquely. I assert when the yellow cells are firing here, the rat is thinking about being in the yellow portion of this maze. When the orange cells are firing, the rat is thinking about being in the orange portion of the maze. Now, isn't that amazing? Let's see that in real time. What we'll see is an actual recording of hippocampal place cells as the rat explores an M-shaped elevated walkway. The position of the rat is going to be denoted by the white light. He's connected to a wire that allows us to record and amplify the neuronal signals from about 10 individual cells. The firing of those cells will be denoted on the left-hand side of the maze. We'll put a little vertical tick mark whenever that particular cell fires. The cells are color-coded, or the tick marks are color-coded, so that we can map the activity of the cells against where the rat is in space. So let's see what happens. I draw your attention to the following. Note how the rat is going first up and then left and then down. That's his path through space. As he negotiates this space, the cells follow a particular pattern. The blue cells first, then the kind of greenish ones, then the yellow ones, then a few reds, and finally the pinks when he finishes. The sequence of colors map to the rat's path through space. Knowing which cells are firing, we can place the rat in space. Knowing the sequence of cell firings, we can map his path. We can use this information to see a rat think. It turns out hippocampal place cells fire at times other than just running through the maze, and that fact is going to help us understand how a rat thinks about a path. That other time hippocampal place cells fire in order is something called a sharp wave ripple. It's only about 1/10 of a second long, but during those sharp wave ripples, we can see place cells firing in sequence: purple, blue, green, yellow, pink, then brown. We know that place cells actually map to space, and if we plotted those spaces, we would see a trace of the path. The sharp wave ripple shown on this slide is the rat thinking about moving through space. Down, right turn, then up. The ripple is only 1/10 of a second long, much too fast for movement to occur. This is the rat thinking. And that kind of thinking is an essential underpinning of deliberative decisions. The other part of the brain we need to talk about before we put it all together is something called the basal ganglia, shown highlighted in this rotating view of your head. The basal ganglia sit in the middle of your brain. There are multiple individual areas, but we don't need to go into them right now. We'll just consider them as a whole. I want you to think about the basal ganglia as the go-no-go system of the brain. Various potential movements or behaviors compete in a race condition within the basal ganglia. The first one that gets to a threshold gets committed to behavior. The other potential behaviors are suppressed. Let's see how that works. We'll consider two potential actions: turning right or turning left. We're going to look at a slightly different metric of neuronal behavior called a heat map. In a heat map, the color relates to how many neurons are active and excited. The warmer the color, the greater the activity. Deep reds mean lots of neurons are firing. Deep blues mean very few neurons are firing. In this diagram, we map neurons in the basal ganglia associated with either turning right or turning left. Each trace is aligned with the arrow that points with time. There's probably maybe 100 or so neurons represented here, and we're following them in time. We see many significant phases. The first is both potential actions show activity early on. Activity builds for both choices until one of the actions crosses a threshold. Once it crosses that threshold, that potential action is committed to behavior. It'll take some time before the behavior is actually expressed. Movement objects have got to be formed, signals have got to be sent out to the muscles, etc. The prime point I want to make here is that 1, potential actions are all simultaneously active. 2, one action gets to the threshold before any other. And 3, That threshold crossing action is then the one committed to behavior and expressed. I also draw your attention to the non-expressed movement. It goes very, very blue. That means that potential action is being suppressed. That's a hallmark of this system. One potential action hits a threshold, it's committed to eventual behavior. The other actions are suppressed. This effect could be important in obsessive-compulsive behaviors, situations where one action is chosen to be done over and over again in order to suppress a different kind of behavior and its associated emotions. But let's keep it simple. Right now, the main points are multiple actions being represented, one hits a threshold for action, and that's the one committed to behavior. Let's put this all together. In this maze, choosing the green path has been consistently paired with food. Turning to the right is the path to reward. No food has been paired with turning to the left, and that's the reddish path. What's going on through the rat's brain as she gets closer to the choice point? Well, her hippocampus is using sharp wave ripples of place cells mentally time traveling down each choice path. Turn right, there's food. The reward system, which can also travel in time, evaluates that thought and produces burst of activity which is fed into the basal ganglia. Her hippocampus will also mentally travel down the left path. No reward there, no burst of activity from the reward system, and that is fed to the basal ganglia. The basal ganglia, as a group, evaluates turning right versus turning left until one action exceeds a threshold. Turning right ultimately gets to the threshold via activity in the reward system and then gets committed to behavior. A movement object will be expressed, muscles will point her to the right, and she'll scamper down that right-hand part of the T maze, and the left-hand turn will be suppressed. Isn't that awesome? Now let's see some of this in actual operation. We're viewing a rat in a self-returning maze from above. He will run down the center of this maze and then make a right or left turn and come back to the top and then run down the path again. The film has been edited to show alternating choices, first to the right and then to the left. We'll look at some cells that are active as the rat is deciding what to do. See if you can use the actual activity of the cells to predict which way the rat is going to turn. Listening to those cells, I hope you reached the conclusion that they were active and bursting when the rat was going to go left. The behavior in these cells demonstrate what we talked about today, the brain making a decision, a deliberative decision. Remember also in the introduction when I said, you think like a rat? Well, sharp wave ripples we see in a rat are also present in humans and a lot of other species as well. Well, it's time for Homer to join us again and talk about the two major points of this session on deliberative decisions. First, Homer, in his own unique way, says that even rats have brains that can travel in time. This turns out to be of fundamental importance in the way to understand our brain. Our brain can move us around in time and space just like a rat's brain can move us around in time and space. Activity from those events can then feed other systems that can determine which particular potential action is actually going to be expressed in the movement of muscles. But Homer also has a second point, and if you know anything about Homer, you know he's a bit of a lazy guy. So what he says is, it sure seems to take a lot of effort in making this kind of decision. And he is very, very right. Your brain takes 25% of the caloric load of your body. In other words, 25% of the energy in your body is spent on the working of your brain. Mother Nature is very careful about how you spend energy, and so that brings us to the fourth kind of decision. Habits. Habits are decisions that your brain has delegated and really not much engaged in anymore. Habits occupy an amazing amount of our time, and it turns out they're actually quite useful. Almost half of the behaviors that you do on any given day are habits. I bet you didn't know it, but there are psychologists who measure how we spend our day, and their research shows that about 45% of the behaviors we do on any given day is a habit. In nature, the percentage for wild animals can be even more. If you don't think you're a creature of habit, try changing your shower routine. Your shower or bath routine is a highly stereotypical behavior for most of us. That means that we really don't deliberate over each individual behavior we're doing in the shower. Each behavior seems to be cued by the behavior just before. First wash this bit, then that bit, then the other bit. We tend to have rigid routines when we bathe, as we drive to work, as we tie our shoes, etc. We are all creatures of habit. The benefit of habit is reduced energy load on the brain. And I mean, think about it. Thinking is energy intense. Just deciding whether to go right or left takes significant processing in the hippocampus, mental time travel, and in the basal ganglia, go or no go. And that's before the muscles even start moving. In fact, when you sum it all up, your brain, again, consumes 25% of all the calories burned by your body. And no, thinking cannot get you to lose weight. Only moving your muscles can get that. But habits do conserve energy. Rather than all your brain being used to deliberate and decide something, just using a part of your brain, actually just a part of the basal ganglia, is used with habits. And that's a good thing, right? Well, as beneficial as habits might be for energy utilization, they're also a source of much human and hamster misery, because habits, once they're formed, are very, very difficult to break. Your brain is on autopilot during a habit. Habits can be maladaptive, and it's very, very difficult to break chains of autopilot behavior, because your brain is really not evaluating other options. It's really not deliberating anymore. It's performing automatic associative decisions where one step leads to the next, and at the end of some of those chains are not good places. So Homer's going to weigh in at the end of this section and give two observations of note. The first is Homer really likes habits. They help his simple brain not get too tired. Homer is not one to really apply himself most of the time. Homer is absolutely a creature of habit. He manages his brain at a very low energy level, and that's just fine for Homer, and we wish him well for that. But Homer also notes that once bad behaviors turn into habits, they're rigid, and therefore very difficult to change. And this is one of the challenges of modern behavior, and I think Homer's got it nailed here. The study of habits and the way of breaking habits can offer great hope to segments of our population who are really in need. I'd like to close our seminar today with some overriding thoughts. First, 3 of the decision systems we've talked about today, reflex, associative, and habit, are all automatic. Your brain is wired to do this. Yes, you can override resources, and there's ways of modifying habit, but in general, these decisions and the systems are automatic. They reflect the way your brains are wired by Mother Nature. So they're not going to change. They haven't changed for thousands of years. Most of the time, say 80% or so, they seem to work well. But when the ultimate behaviors out of those systems are ill-suited to the facets of modern life, it's not necessarily because people are evil, it's because their brains are wired that way. To make progress, we need to switch to manual mode. This is a deliberative choice or decision to override what our automatic brain systems are doing. We can use these deliberative systems to address automatic biases and other kinds of associations that our brains naturally make. One example comes from modern symphonies. People generally tend to pick groups that are similar to themselves. In other words, us groups like other us groups or in groups. So if white males already in the symphony choose auditioning players, white males are going to tend to be the ones that white males pick. Modern symphonies have found that blind auditions accompany and address such an automatic bias. Performers audition behind a curtain, and therefore the evaluator doesn't know the gender, doesn't know the race, doesn't know the ethnicity of the performer. This has turned out to be a wonderful way of eliminating gender and other kinds of bias in modern symphony. The insights of blind auditions is sufficiently powerful to have achieved the Nobel Prize for the economics who first studied these effects, Claudia Goldman, and good for her. A similar process is now being used by NASA to allocate time on the Hubble Space Telescope to eliminate gender, ethnic, and university bias. And finally, this process is being tested in the legal system to address hidden biases based on race and neighborhood. The key thought here is to use our deliberative systems in our brain to override our biased brain. An afternoon of training is not going to eliminate the evolutionary skill hundreds of thousands of years old. Only other parts of our brain, devising new modes to counter this national bias, is going to address the situation. I'm glad we're starting to make real progress using these approaches. This concludes Seminar 2, The Decision to Act. I hope you enjoyed it. Let me foreshadow the topic of Seminar 3, The Structure of Memory. Memories are complex, multidimensional, and slippery concepts that have several processes within the brain. They also have a structure within the brain. We'll talk about in the next seminar what those essential characteristics of structure are. One type of memory is like a story from Proust, the French writer. Proust wrote 7 volumes and thousands of pages about a memory that came from a fateful encounter with a famous Madeleine and a cup of tea on a cold winter's day. His memories came out in rich, detailed pattern. However, this is but one way memories are structured. Are there others? Well, yeah, and there are different brain structures which bring them about. A good analogy is the totem pole of the Pacific Northwest Indians. Totem poles are not like an elaborate reconstruction of a story as told in the pages of a book. They're essential elements of the story as carved on a totem pole. To give a familiar example, the totem pole of Goldilocks and the Three Bears would be a pole that has a bear, a bowl of porridge, and a bed carved on it. The story would be reconstructed from these symbols. Now, of course, that means that 2 different people might tell the story in 2 different ways, or the same person on 2 different occasions might tell the story again in 2 different ways. We'll see how memory structures relate themselves to brain structures in underpinning Proust-like stories and Pacific Northwest totem pole stories. Please join me next time to see the fascinating story of the structure of memory. This is Steve Saltwick. See you next time.
Ciégame, mata mi corazón. En nuestra alcoba azul